Right. In the first part of this lecture series, we introduced the concept that there were these modes of inheritance that could be affected by or conditioned by your sex, whether you're male or female. And we talked about two in particular, and these were sex limited and sex influenced. And we said those were autosomal, right? They're the ones they're found on most of the common chromosomes that we have. And I mentioned at the time, I said, okay, these are autosomes, right? These are where everybody gets two copies. You get one allele from mom, one allele from dad. Everybody has them, whether you're male or female. And then the system really kind of determined by hormonal control and other things how those actually get finally expressed, what the final outcome of those. Sex limited, we said, was limited to one sex, right? And sex influenced, while both sexes would have the trait, they wouldn't necessarily express them in the same exact way. And we used the example of male pattern baldness for that, and saying that even for dominance in males, it was recessive in females. So you had to take two copies for females, and even then, they don't express the full, often express the full exposure of dominance, which baldness. Okay. Now, what we're going to do now is we're going to move over to looking at what happens when you have things. The particular loci are actually carried on the sex chromosomes, whatever types of sex chromosomes you have, right? And how that differs from things that are autosomal. So before we do that, before we actually get to that particular system, we're just going to think about what sex chromosomes are, okay? And we're going to talk about some what we call modes of sex determination. And we're just going to kind of review a couple. There are actually multiple ones out there in systems. We're just going to kind of look at the big picture of a couple of examples that are going to be important to us. But we're going to concentrate now on the sex chromosomes. So these are specialized chromosomes that are somehow involved in sex determination. So let's think about sex determination itself. Well, the first of the two big models we want to look at is one that's called the pro-tenor mode, and it's generally abbreviated as XXXO. In this case, the O means absence. There's no chromosome there, okay? So essentially what you have is you have a sex chromosome. If you get two copies of that sex chromosome, which we're calling X, then you're one sex. If you only get one copy, you're the other sex. Frequently in biological systems, when you only get one copy, you turn out to be male. And there are some cases of fishes and things where that's the reverse, but for the most part, getting two copies of the same sex chromosome, being a homozygote for that, and having them is going to make you female. Getting one copy, being what's called hemizygous, is going to make you usually a male. Now, this works out fairly well because, with sexual reproduction, we cross a male times a female. So essentially what you're always doing is crossing a homozygote, if you want to think of it that way in terms of sex, okay, not necessarily the alleles that are carried there, but as far as the ability to produce one sex or the other, it's homozygous, times a heterozygote, right? And when you do that, if we just do a little Punnett squares to make sure we can see what we're doing here, you expect about half your progeny to be female in this case because they're going to get an X from mom and an X from dad. And you expect about half your progeny to be males because they're going to get an X from mom over here, but they're going to get nothing from dad. So they're going to be missing that particular chromosome. So this ends up producing a 1-to-1 ratio of males to females. This is known as the primary sex ratio. Okay? So primary sex ratio is what you expect at fertilization. Now, there's the secondary sex ratios that we'll talk about later, but secondary sex ratios can be very different than the primary ratio, right? In changing over to humans, which have a different sex determination, but in human systems, it turns out that there's more females, that while there are potentially more males actually fertilization, more females make it to birth sometimes. This is true in other species. Certainly as we age, right? The secondary ratio can really change in humans. If we were to go look around a university, it's pretty much even, right? It's relatively 50-50 male, female by that point. If we went to some place where there's a lot of retirees, maybe, somewhere in southern Florida or even retirement communities, there's probably many more women there than there are males because males tend to die off earlier. So if you look at the secondary sex ratio in a group of people that are 90 years old, probably see that there are more females than there are males. And the same thing happens in natural systems that are not with humans. But for here, one of them makes a male, one of them makes a female. It's a primary ratio you expect. Just some examples, crickets. The fundamental number, the two-in number of crickets, different species can range widely from somewhere around 7 or 8 up into the 20s. But what's important for us is that the males will always be an odd number. So if they only have 7 or 8 chromosomes, the males will have 7, females will have 8. If they have 14, the males will have 13, the females will have 14. And so there's good examples. And you actually see problems in textbooks and things that deal with crickets. And when you see a cricket and it says, oh, I've got my pet cricket, Billy, and Billy is, I know he's male. Why? Well, because he has an odd number of chromosomes, okay? All right, now let's go and let's think about human systems and mammal systems. Mammal systems, instead of being under the pro-tenor mode, fall into what's called the ligase mode. Ligase mode is different in that you have 2 very distinct sex chromosomes, okay? So remember, pro-tenor mode, there's only one kind of sex chromosome, and you get 2 of them or you get 1 of them. Here, we've got this sex chromosome, and then we've got some other odd sex chromosome over here, usually smaller, okay, sex chromosome. That's the ligase mode, all right? And we'll talk about why we write them down as XX or XY or ZZ, ZW in just a moment, right? This is an ideogram, and we'll talk more about those when we talk about chromosomes, but again, this is just an alignment of human chromosomes. It could be any species. This happens to be humans. Humans are classically part of this ligase mode, and they have in the males, you have an X chromosome and a Y, and they're very different. One's a big X and one's a small Y. Okay, and females, of course, get 2 X's, right? That's the way it works. We get so it's odd numbers of chromosomes. Okay, now we were not going to spend too much time worrying about this, but just to let you know, because everybody gets an X, all mammals are directed toward being female from the start. Okay, and that's due to the fact that there are these stride genes and other genes that come into play, and if those are absent, okay, then you just direct line toward being female. You need something to divert you off to be male, so the stripe is present, which is associated with the Y chromosome in mammals, and once that's expressed, then that individual the organs will turn off and direct themselves toward production of testes and the prostate and other organs of the primary sex organs. But we're all started out to be females, and it just takes a design the other. And there's quite a few mutations that can occur that can change that. We'll talk later about these, but this AR means autosomal. And so what I'm showing you here is that there are a number of, not only is it the striate locus, but there are lots of different loci that once this kicks into play, they all have to work to make sure that these initial structures, these gonopodial ridges actually eventually end up either being ovarian tissues or they end up being testicular tissues. Okay? And then there's many mutations. Here's an autosomal dominant here, autosomal dominant recessive. Here's an X-linked one that we'll talk about in a moment. Lots of other autosomals. Point of this is while there's a kind of a driving chromosomal product, okay, so a driving gene that starts the whole diversion. Once you start that diversion to male or female, then a whole bunch of other genes, suites of genes turn on to actually formulate the final production of that product. And you can have mutations in all of those, and when you do, you have all sorts of outcomes from it that end up being producing transgender and producing individuals that have both partial reproductive organs and all sorts of things. All right. But get back to talking about this, all right? Now that we have 2 different sex chromosomes, we have this one and this one, okay? Individuals that end up with those are called the heterogametic. Hetero is like hetero means different, right? So these end up XY or Z, or called ZW, are the ones that are heterogametic. So males like me and humans or mammals are the heterogametic sex. Okay, and the reason we have ZW here is what we're going to show you next, right? The homogametic sex is the individual where you end up with both the chromosomes being the same. In mammals and humans, this is the X chromosome. Individuals that are homogametic both get the X chromosome. In many fishes, different types of fishes, moths, butterflies, and all birds, the individual that's homogametic, that gets the two things, is actually the male, right? So that's why we use what's called the Z system, the ZZZW system, to indicate that it's backwards from what we're thinking about in mammals. So the individual that's heterogametic in birds is the female, and the individual that's homogametic in birds is the male, which is completely opposite of what we see in mammals and humans, okay? Very important to remember that. And to point out a couple of things, okay? One, penguins are birds, okay? Why do I say that? Because I've asked on tests, and people somehow think penguins are fish or something. I don't know what they think they are, but they're birds, which means that a male penguin is the homogametic form, and the female penguin is the heterogametic. Now, the females still produce the eggs and do all the other things that that we see in mammals as far as the female, but they just have a different genetic system that pushes them toward being male or female. And I'll also remind you, kind of silly, that birds, all birds, including chickens, are this Z system, okay? Invariably, when I teach a face-to-face class, I will give a test and a hand will go up and somebody will say, I can't figure a problem out. The reason they can't figure the problem out is they're trying to work it and they don't realize that the female is heterogametic, okay? And so they'll struggle. But just remember, birds, moths, butterflies, you need to reverse it into the Z system, okay? All right, there's one other term we need to talk about, okay? That term is hollandric, okay? Hollandric, and particularly related to mammals, is the information that's carried on the Y, right? So you have this big X chromosome and then this little Y chromosome, right? Well, obviously you're not going to put really important genes on the Y because only half your population gets it, right? Okay, you put a lot of information on the X. X has lots of loci besides determining sex or anything. The Y has some things about driving determination of sex, a little bit of testicular development stuff, but most of the things that make us male, most of the loci are actually autosomal. They're off somewhere else, okay? And you saw that from that big list of mutants where many of them said autosomal recessive, or autosomal dominant, right? Very few of them indicated they were on the Y. But we do pass things down hollandrically, okay? Which means that they're passed from father directly to sons. No daughters are going to get them because they don't get the Y. And the word for that is hollandric, okay? So be sure that you've kind of underlined that term and know what it means. All right, now just to go back and show you that the primary sex ratio, just like it was for the pro-tenor mode, which was, is XO, well, here we've still got a het over here, technically. We've got an XY male with a female who's XX. And if you just do anything with that, you're going to get half your progeny, going to get an X from both parents. Half your progeny, you're going to get an X from one parent, but a Y from the other. And so you're going to get a ratio, primary sex ratio of 1 to 1. Okay? Again, I said earlier with the pro-tenor that that's the primary ratio. Turns out that Y-bearing sperm are slightly smaller and faster, and they can, and X-bearing sperm are slower and bigger, and so depending on when the intercourse occurs and the timing to ovulation, you can have different things, different ratios, but mechanically, we ought to expect about 1 to 1 ratio of males to females. Okay, all right. So that tells us about sex determination, okay? So now we're going to come back in the next lecture and we're going to talk about, all right, once you have these sex chromosomes and you have different ones, you have some people getting one sex getting double copy and one sex only getting half a copy. What does that do to the mode of inheritance? And that mode of inheritance that's involved there is called sex linkage by loci that are actually carried on the X, or in the case of the Y, but again, the Y is not very important here. We'll be talking about almost everything that's carried on an X and how it differs between males and females. So that'll be in the next set.